Okay, class, today we're looking at a microscope. The first thing that I want you to know is how to hold the microscope. When you're carrying it, put your hand under, hold it like this to support the microscope so it doesn't fall. It has a power cord you plug in, and there is a power switch on the side of the microscope. Sometimes it's on the side, sometimes it's in the front. This one happens to be in the front here, but there are lots of different types of microscopes. And now we're going to go through the parts of the microscope. Okay, we are going to work our way from top down on this microscope. This is a compound microscope. First you have the ocular lens or the eyepiece that you put your eye up to. This is the body tube that holds the ocular lens. And in this case, this ocular lens has in itself a 10 power. There's an adjustment here for making sure not all people's eyes are created equally, so there's an extra adjustment there. But again, this is 10 power, so just looking through this alone does that magnification. This is the revolving nose piece. It turns, and you see this shortest objective here is a 4 power. So if I was to start looking at my slide with a 4 power, times 10, I'm looking at it 40 times power. So each one of these, from progressively working our way down, we have a 4, we have a 10, we have a 40, and we have a 100. So we would be going from looking at an overview of a cell to looking at the internal parts of a cell with these other objectives. This part that I carried is called the arm. This is the stage, and within the stage is the stage clips. And so when I have a slide with a prepared specimen on it, you have a slide, which is a piece of glass. You have a cover slip, which covers that. And if I did a cross-section of a plant material, I'd put the cross-section on there, a very thin slice, and then I would use deionized water to put a drop of water over it, and then I would cover it with the cover slip. It makes a translucent thing that you could see through, and you would put that on the stage, and then what happens is these stage clips hold, and you can do fine adjustments and coarse adjustments. So fine adjustments and coarse adjustments are to move the stage forward and back. See, I'm just rotating each one of these forward and back. We also have the coarse adjustment knob for looking at your specimen. So this is a coarse adjustment, and so by turning this, I'm bringing the objective down to the slide, and when I get the view in the ocular lens that I like, then I can do a fine adjustment to really focus it in. All right, we have a light source and a base that holds it. The light source oftentimes has different diffusers. This has a diaphragm diffuser that can cut the light or increase the light. If you see there, the light coming through the slide, so when I have that and I'm looking in there, you don't want to be blinded, but you want to have enough bright light that you can see through your specimen. So that's our light source. Compound microscope, dissecting microscope. This dissecting microscope has the ability to move up and down, right or left-handed, and to look at a specimen without magnification into the cellular level. We would just be looking at something, for instance, if we were looking at a flower structure and we wanted to have a a 3, 4, 5 times the power magnification to see better into there. If we're looking at an insect and we wanted to see the legs, or if we're looking at the stem of a leaf, the petiole, and we wanted to know if it has hair on it or not have hair structures on it, this would be the way to do that. That's a dissecting microscope. A lot of times a specimen is laid on there, you work on that, and at the same time you are working through here. We do this a lot with our Venus flytraps when we're trying to cut out very fine pieces and get structures out from our Venus flytrap. We would look through a dissecting microscope. Kind of a big picture view and kind of a dialed in view with a compound microscope.